All right, if you'll recall, last time we were looking at what chlorophyll can do all by itself, how it's able to absorb energy and release that energy, and how it involves the excitation of electrons. And those electrons are key to what happens during the light-dependent reactions, and we'll be looking at that in greater detail today. But again, to orient you as to where this happens within a plant cell and more specifically within a chloroplast, we're going to go ahead and identify the different components of the chloroplast so you understand very clearly where everything takes place. So of course here we're looking at a leaf. We're zooming in on the plant cells that make up that leaf, and those plant cells, as you can see, have a cell wall around them, and within the cells, all these little green things, basically everything that you can see within the plant cells, are chloroplasts. So we're going to go ahead and label all the structures of the chloroplast on this diagram because they're all visible on the diagram, and then we'll relate that to this transmission electron micrograph of a real chloroplast so you understand what you're actually seeing there. So this whole thing's a chloroplast, of course, and then if you'll recall, chloroplasts are one kind of a whole family of organelles known as plastids, and they're all double membrane bound, if you'll recall. They all have two membranes around them. So the chloroplast has an outer membrane and an inner membrane. The space within the chloroplast, and I think I've told you before, you can think of that as kind of like the cytoplasm of the chloroplast, but we don't call it that. It's known as the stroma. And then there are these stacks of discs. Each individual disc, as you already know, is called a thylakoid. And the whole stack of discs, the whole stack of thylakoids, is referred to as a granum, but collectively they would be called grana. In other words, plural would be grana. And then if we take a look at the individual thylakoids and we cut them open, you can see that they are actually hollow inside. But before we look at that, let's take a look at how this relates to the transmission electron micrograph. So what you're seeing here, these dark areas within the chloroplast, are the actual grana. That's what you can see when you look at real chloroplasts with a transmission electron microscope anyway. So if those are the grana, and basically everything that you see in here is thylakoid membrane, then that makes the space surrounding it the stroma. So back to the diagram, one individual thylakoid is surrounded by a membrane. And so that's what's referred to as thylakoid membrane. You'll notice that the thylakoids are also connected to each other between grana. So all of that is made up of thylakoid membrane as well. And inside the thylakoid membrane, because they're hollow, or inside the thylakoid I should say, because they're hollow, that's referred to as the thylakoid space, also referred to as the thylakoid lumen. Lumen's a general term, referring to the space within anything. For example, your stomach, where your food goes when you swallow your food into your stomach, the space within your stomach is referred to as the lumen. So lumen is a general term in biology that just refers to a space, but we're going to use it in the future, so you do need to be aware of that term. So this is the second time that we've looked at the structure of the chloroplast. And so obviously it's important for you to understand the structure and understand where the different parts of photosynthesis take place. So we're going to point that out now before we move on. The first phase of photosynthesis, if you'll recall, is referred to as the light-dependent reactions because they don't function without light. And if you'll remember, they take place in the thylakoids. Okay, so the thylakoid membrane is responsible for absorbing light energy. That's where the light-dependent reactions take place. The second phase, the light-independent reactions, also known as the Calvin cycle, occur in the stroma. So they occur in that fluid-filled space that surrounds the thylakoids, or surrounds the thylakoid membrane within the chloroplast. So that means that the thylakoid membrane is where you can find pigments to absorb the energy of light, like chlorophyll. So the pigments are found in the thylakoid membrane, making up the thylakoids, because that's where light energy needs to be absorbed in order to carry out the light-dependent reactions. So if we take a closer look at the thylakoid membrane, this is what it looks like. So this diagram is taking us from the organelle level to the molecular level. So notice we're seeing individual molecules here. That's why I'm referring to this as the molecular level. It's still a diagram. Unfortunately, we really can't see molecules at this level of detail using a microscope or anything. So we're forced to rely on diagrams for this kind of thing. But of course, this represents a chloroplast, and this is the inner and outer membrane, of course. The stroma surrounding the thylakoids here, the grana, the stacks of thylakoids. And so that's one granum, and then this is one thylakoid, just to make sure you're oriented here to the diagram. And what we're zooming in on here is one piece of thylakoid membrane from one thylakoid. And so hopefully you'll recognize that as being made up of what all biological membranes are made up of, phospholipid bilayer. But that's the thylakoid membrane. So all these individual molecules that are making up the thylakoid membrane are phospholipids, if you'll recall. And also, if you'll recall, just because it's important for you to know, is that those molecules have a hydrophilic end and a hydrophobic end to them. And that's why they arrange themselves into this phospholipid bilayer. They pretty much do that automatically when they're placed in water, and it's their interaction with water that causes them to do that. That's review. The important thing we're looking at here is where the pigments are, and that's what this thing is that's embedded in the phospholipid bilayer. And as you can imagine, anything that's embedded in the phospholipid bilayer is typically going to involve protein. So this is a complex that's a combination of protein and pigment molecules. So this is known as a photosystem. So it's proteins plus pigments. So each one of these green discs here in the diagram is representing a chlorophyll molecule, but it could be other pigments also. There can be other accessory pigments within the photosystem along with chlorophyll to absorb different wavelengths of light. So a photosystem is a light capturing unit that's embedded in the phospholipid bilayer of the thylakoid membrane that contains the pigments to absorb the light energy for the light-dependent reactions of photosynthesis. And there are two types of photosystems. We're only seeing one photosystem here, but there are two types of photosystems, and they work together during the light-dependent reactions, and they are known as photosystem 1 and photosystem 2. And they were named in the order of their discovery, which is about to become somewhat confusing. And don't freak out when you see the next slide. We're going to take it one step at a time, so bear with me. So here we see the process that's going on in the thylakoid membrane. And like I said, don't try to rush ahead and fill this out, and copy it all down, and unless you want to copy it all down and then come back and listen to my explanation. But if I were you, I would add it as I explain it, and if you need to pause, go ahead and pause. So the first thing that I want you to understand is the light-dependent reactions. A process takes place during the light-dependent reactions known as photophosphorylation. And we'll come back to that term when we're all done here so you can see what photophosphorylation is referring to. But let's start by identifying the structures and the spaces that are involved in this process. So this whole thing represents the thylakoid membrane. So that's the first label that you should add, is thylakoid membrane. So we're seeing the phospholipid bilayer, and you'll notice that there are proteins that are embedded in the phospholipid bilayer, and two of these, the big green spheres here, are representing photosystems. So you want to label this one photosystem 2 and this one photosystem 1. And so what's confusing about that is that photosystem 2 operates before photosystem 1 during the light-dependent reactions. So don't let that confuse you, but you need to remember that. in between the two photosystems is a proton pump, what we're going to label a proton pump. And remember, proton is another term referring to hydrogen ions. So it's going to pump hydrogen ions across the thylakoid membrane. Also embedded in the thylakoid membrane is a protein that is specialized to form ATP, to add a phosphate onto ADP to form ATP, and that's known as ATP synthase. And again, don't worry about putting any of this other information on there. Just label the thylakoid membrane, photosystem 2, photosystem 1, the proton pump, and ATP synthase at this point. But you can, so the next thing we're going to label then is the stroma. You know, we need to know which side of this thylakoid membrane is which. What space is on which side here? So the stroma is down below. That's outside the thylakoid, if you'll recall. And then there's the thylakoid space, also referred to as the thylakoid lumen, that is being represented up above the thylakoid membrane in this diagram. So now that we know what and where everything is, we can take a look at the process. And the process, of course, begins with light energy being absorbed by pigment molecules in the photosystem. So chlorophyll embedded in the phospholipid bilayer here, forming photosystem 2, will absorb light energy in the form of photon, and that excites electrons. As we saw in chlorophyll, electrons get excited and they get picked up. So they don't fall back down to their ground state and emit fluorescence, emit light and heat. When they get excited within the photosystem, they get picked up from the photosystem by these electron transporting proteins in the thylakoid membrane, and they get transported through the proton pump, and then there's another electron transporting protein that picks it up on the other side of the proton pump and transports it to photosystem 1, where photosystem 1 also has pigment molecules that absorb light energy in the form of a photon, and that further excites the electron, which is ultimately passed through another electron carrier to and an enzyme that adds that electron or delivers that electron, I should say, to NADP+, and along with a hydrogen ion forms NADPH. And that is chemical energy. That's one of the products of the light-dependent reactions, the important products of the light-dependent reactions. It's an electron carrier, and it's going to carry those electrons, that's a form of chemical energy, to the Calvin cycle, to the light-independent reactions, where carbohydrates are going to be made. So this chain of proteins in the thylakoid membrane that transport electrons is referred to as an electron transport chain. And interestingly, we're going to see an electron transport chain when we study aerobic cellular respiration too. So don't get them confused. There's an electron transport chain in both processes, and they both are functioning in a similar way. So now we need to come back and see why it is that plants need water and split water at the beginning of the light-dependent reactions. So you'll notice water here is being split to free up hydrogen ions, and in the process, oxygen is being evolved. There's an oxygen evolving complex that has enzymatic activity that is responsible for this. But what's going on here is that the plant needs to split water both to get the hydrogen ions, but also to get the electrons to replace the electrons that left chlorophyll and other pigment molecules. So that electron that got excited within chlorophyll and picked up and that flowed and moved along the electron transport chain has to be replaced. And so the electrons are replaced by electrons from water, so that this process just can continue going. As long as there's water to supply electrons, those electrons can flow through the electron transport chain and power this process. And along with the electrons, the hydrogen ions are important because they're going to go on to the Calvin cycle to supply hydrogen to become part of carbohydrate. But some of those hydrogen ions are not going to stay in the stroma outside the thylakoid, because the electrons that are passing through the proton pump are powering that pump to pump hydrogen ions, to pump protons from the stroma into the thylakoid space. So we end up getting a high concentration of hydrogen ions, of protons, in the thylakoid space, especially compared to the out in the stroma. So there's a relatively low concentration of hydrogen ions out in the stroma. In other words, what has been set up here is what's known as a concentration gradient. Hopefully you recall that back when we were studying diffusion and osmosis. So there's a very strong concentration gradient for hydrogen ions, for protons, high concentration in the thylakoid space, low concentration in the stroma. So those protons want to, so to speak, diffuse across the thylakoid membrane or through the thylakoid membrane. However, they can't go through the thylakoid membrane itself, the phospholipid bilayer, because of their positive charge. So the only way they're allowed to go through is through this special protein, and it is a very special protein known as ATP synthase. And the energy of that diffusion of the hydrogen ions is used to power ATP synthase to add a phosphate onto ADP to make ATP. So this enzyme, as the protons diffuse through it, the energy of that diffusion is used, and this thing actually spins. You'll notice there's an arrow here showing it spinning. The energy of those protons coming through the ATP synthase causes it to actually spin. They've shown this experimentally. And that spinning, that mechanical motion, actually the energy of that is used to add a phosphate onto ADP to make ATP. And this just constantly runs. As long as there are protons within the thylakoid to diffuse through ATP synthase, it will continue to make ATP. This process of diffusion of the protons is known as chemiosmosis, or chemiosmosis, however you want to pronounce it. So it's got a special name. Because it's such a special process, it's got a special name. And this is, along with the electron transport chain, this is another process that we're going to see again functioning during aerobic cellular respiration. So that concept of chemiosmosis goes along with both photosynthesis and aerobic cellular respiration. And that gives you some idea of why we're studying these two processes within the same unit together, because there are a number of commonalities between them. But this whole process, starting with light energy and ending with the phosphorylation of ADP to make ATP, is the reason that the light-dependent reactions are also known as photophosphorylation, because ATP is being phosphorylated using the energy of light, photo, right? So photophosphorylation is what's happening during the light-dependent reactions. So ATP is the other important product of the light-dependent reactions. So both NADPH and ATP and hydrogen are going to go to the Calvin cycle and participate in forming carbohydrates. So let's put this in writing here. So the light-dependent reactions use light energy to split water, releasing oxygen gas. That's where the oxygen comes from. It's a byproduct of the light-dependent reactions. Photon-excited electrons, right, from chlorophyll, but replaced from water, flow through the electron transport chain. So there they are, flowing through the electron transport chain from photosystem 2 to photosystem 1, which doesn't make a whole lot of sense, but that's the way they're named. And that supplies energy to pump protons into the thylakoid space. So from the stroma into the thylakoid space. And electrons are finally accepted by NADP+, along with hydrogen, to form NADPH. And that's a major product here, NADPH. Protons then chemiosmose into the stroma through ATP synthase. Protons chemiosmose through ATP synthase into the stroma. And in the process, they can convert ADP to ATP, or you can also say they recharge ADP to ATP, the other important product of the light-dependent reactions. So both NADPH and ATP are supplying chemical energy for the production of carbohydrates in the Calvin cycle. And that's where we're going next. ...